Oftentimes I like to ask my students if they think that a cell is more complex compared to a 747, let's say. And of course we do not know the answer to this question, but the answers I get from them, they are about 3-to-1 in favor of a cell. The difference, of course, between a cell and the 747 is that we have built the 747, so we know how it works, and if we don't understand the part of it, then we can just go there, dissect it, and find out what's going on inside that system. We could not do that with cells until very recently. This is all changing in recent times because of the unprecedented ability that we are developing to go inside the cell and measure a lot of different components of the cell. And of course, as a result of that capability, we can now ask the question how all of these pieces come together and how do they work together to give us the functions of the cell and the cell in connection with the bigger organism that we try to analyze. In the context of biotechnology, the two organisms that we are studying at this time are Escherichia coli and the photosynthetic bacterium Synecocystis. In E. coli, we try to understand the mechanisms that allow these cells to accumulate large amounts of lycopene and beta-carotene, and then as a result of that understanding, to make the cells produce more of that useful chemical. One technique that's being used to allow us to visualize the activity of genes such as these genes is to couple the expression of these genes with a marker such as green fluorescent protein. And if you see a large amount of fluorescence, that means that there is a high degree of expression of the gene that is involved in the synthesis of lycopene. For the case of Synecocystis, Synecocystis is a photosynthetic bacterium. It has the ability to fix carbon dioxide from the atmosphere, and it also has the capability to convert that carbon dioxide into a number of different valuable products. One of these products is a biopolymer, and what we try to do in the lab is basically two things. One, to accelerate the rate of fixation of carbon dioxide, and the other to accelerate the conversion of that fixed carbon dioxide into biopolymer. Some practical applications that may come out of this research are the production of biodegradable polymers. These are polymers which will degrade in the environment naturally, leaving no trace behind them. On top of all of that, of course, we are reducing the amount of carbon dioxide in the atmosphere. So all of these products come free from carbon dioxide that we would like to remove in the first place. So the environmental impact is double in this regard. One characteristic of this area is the intense collaboration of scientists from different backgrounds and with different interests, because a lot of activity is taking place at the interfaces between engineering and life sciences. This cannot be done in a single laboratory, and it is important to get many people together in order to answer a particular question.